So welcome everyone. Welcome to this EVOLTREE Online Seminar series. It's the second episode today with Emily Josephs. We are very pleased to have you here, Emily, and thank you so much for joining this series, especially today, which is such an important day in the US, full of emotions. I will not be talking about politics today, but just acknowledging the fact that you're with us on this important date. So just before we start, I wanted to say that the webinar is being recorded and it's also being streamed to YouTube, and it will be available afterwards on YouTube for anyone who wants to watch. Without further ado, I would like to hand over to Antoine Kremer, who is kindly moderating this series and who will say a few words of introduction about EVOLTREE and introduce Emily today.
Okay, hello, hello everybody. My name is Antoine Kremer. I may be repeating what I was saying last time, but I will briefly say a few words about EVOLTREE, who is organizing this series. Now, EVOLTREE is a European initiative, a network of labs with strong interest in genetics and evolutionary research in forest ecosystems, focusing on trees and their associated species. In total, we are about 28 partners from 19 different countries, and in total, this represents about a few hundred scientists with expertise in genetics, genomics, ecology, and evolution. Now, the network is about 15 years old now. It was originally supported financially by the EU, and now it is self-supported by the different partners, even by financial support of the different partners or by in-kind contributions. So what are our objectives? We hope to coordinate and share research efforts and resources at the European continental scale. And what kind of activities are we doing? We build and maintain common infrastructures, for example, field monitoring plots and interoperable databases that are shared between different servers. We organize training sessions in the form of workshops and summer schools and nowadays webinars. We provide financial support for small-scale research projects, and we maintain also, of course, networking activities and edit different diverse dissemination material. So far as introduction for EVOLTREE. Now today is our second session, and we are pleased to welcome Dr. Emily Josephs. Thank you, Emily, for having accepted to our invitation. Emily is Assistant Professor of Plant Biology at Michigan State University. She received her PhD at University of Toronto with Stephen Bright and John Stinchcomb, working on evolutionary genetics of gene expression in Capsella. And she was a postdoc at the University of California with Graham Coop and Jeff Ross Ibarra, working on adaptive differentiation in structured population of maize. Emily's interests address the understanding of evolutionary forces shaping genetic variation within and between domesticated but also natural species and population, and at different levels, from genes, gene expression, and also phenotypes and traits. This includes, of course, questions regarding the maintenance of genetic variation in natural or domesticated population, questions related to local adaptation, and questions related to GE interaction. Her approaches combine population genomics with more traditional quantitative genetics, and her preferred study species are maize and Capsella grandiflora. Now, if I hadn't mentioned her study species, you would have thought that I am introducing a forest geneticist. Emily's research topics are indeed very similar to our own concerns on our own species and our own ecosystems. And I'm sure that her talk will be very attractive to our today's audience. So please, Emily, I'm giving the mic to you.
Just before you start, Emily, sorry, just wanted to say that the presentation will be for around 45 minutes, and then we will have some time for questions. So please save your questions for after the presentation. So over to you, Emily. Thank you.
Okay, thank you so much for the introduction. Can you see my slides okay on the shared screen? All right. Well, thank you so much. And thank you again for the introduction. It was really great. I was really honored to be asked to speak to you all today. As you both mentioned, it's like quite a day. So hopefully we will make it through together, and the news when we finish with this will be really wonderful. So today I have changed a bit about what I wanted to talk about, because I realized that based on the topic of the symposium, I should really focus on thinking about what I've worked on and telling you about what I've worked on related to polygenic adaptation in plants. So there will still be gene expression, but with a focus on thinking about questions related to polygenic adaptation. And because this is a polygenic adaptation symposium, I don't need to spend a ton of time motivating us for thinking about why this is important. But I will say what motivates me as a scientist is just trying to understand what shapes variation within species, whether we're thinking about natural populations of species or domesticated species. And these can range from tons of different phenotypes like phenology or color or size, or even other things that are harder to photograph like gene expression, but might still be quite interesting. And I find it really useful to think about questions about adaptation in sort of a timescale approach. So first we can think about the past. How has adaptation in the past shaped the genetic variation we see today? So for example, when we see different mouse coat colors, I've taken this photo from Hopi Hoekstra, we see different coat colors for mice. What role does local adaptation play in shaping this phenotypic variation and in shaping the genetic variation for these phenotypes? Next, we can think about how organisms adapt to new environments in the present. And there are a number of really beautiful examples of how organisms can adapt to different environments in real time. And so I'm illustrating this with a photo of Trinidadian guppies and these famous experiments where Endler took guppies from one area and put them into a new environment with different levels of predation, and showed that you could get quite rapid adaptation in coloration. But these kinds of experiments have also been done in plants, particularly in plants that are grown by breeders, where breeders are trying to affect an adaptive change, as well as by my neighbor across the street when I'm at work at MSU, Rich Lenski, who's done these amazing, really long-term experiments looking at adaptation to new environments in E. coli. Finally, the reason why it's really important to understand adaptation in the past and the present is that we really want to know how organisms will adapt to future environments, especially as humans, we continue to change the environments in a lot of different and really dramatic ways. And so I've illustrated this with a picture from Rachel Bay's work using a combination of genomics and sort of information about the landscape to try to predict which populations need to change the most genetically in order to cope with climate change. But there's also a large body of work asking these questions using quantitative genetics approaches. But in both cases, really understanding how adaptation occurs is important for thinking about making these predictions. And so today I'll tell you about two different stories looking at polygenic adaptation, or looking at adaptation in quantitative traits, or polygenic adaptation. First, I'll talk about this past perspective, trying to understand how adaptation has contributed to quantitative trait variation in maize. And so answering this will both involve talking about adaptation in maize and talking about the methods development we need to do to even really ask these questions. Next, I'll tell you a bit about a project in my lab looking at how plasticity might contribute to adaptation to herbicides in the weedy species Morning Glory. And so I'll start with this first question. And so the reason why we can think about detecting adaptation and why this is a complicated problem is that detecting adaptation is really complicated. So one traditional way of doing this has been to use reciprocal transplant experiments. So I'm illustrating this with an example of another one of my colleagues at Michigan State, David Lowry, who's done a lot of reciprocal transplant experiments with inland and coastal monkey flowers, or mimulus, where they take populations from these coastal areas, plant them in inland areas, and vice versa, and see this signal of adaptation where coastal populations have higher fitness in coastal regions than the inland populations, and inland populations have higher fitness in inland areas than coastal populations. And this is a really powerful way to detect adaptation. It's been used for quite a long time. However, it depends on having a number of things in your experiment. You need to have a system where you can easily transplant your populations, where you can easily measure fitness, and where you think that fitness will be really similar year after year. And so there are a number of reasons why we might want approaches to detect adaptation that don't require this framework. And I should note, since this is a symposium focused on trees, there are some really cool provenance trials done in trees that are much more varied than this two population system. And I've always thought that's a really cool thing about tree evolutionary biology. So another way to get at adaptation is to look at genetics and try to understand that whether the role that adaptation has played in the past using sort of genetic variation and genomic sequence information. And so this can be really powerful, especially if we're thinking about Mendelian or simple traits. So you could imagine taking a bunch of sequences from human populations and marching through the genome and looking for places where you see big differences between two populations. And if you come across something like this, where when we compare Yoruban humans and European humans, we see a really strong allele frequency difference, where an allele is much more common in Europeans than in Yorubans, this would be a really clear signature of adaptation, because it's unlikely that neutral forces like genetic drift could create such a strong difference in two populations that are relatively closely related to each other. And so in fact, this is the frequency of the lactase persistence allele, which we expect to have had some sort of adaptive effect in human populations. However, these kinds, and I should say that this allele frequency difference is one of the many ways that we can use genomic variation to understand adaptation, including there are a lot of additional approaches. But what they all share is that they don't work as well when we think about quantitative traits. And so we could make the same kind of plot, but for human height, using a bunch of alleles that were found in a genome-wide association mapping study. And now it gets a lot more complicated, because instead of having one clear shift, we see what looks kind of like a big pile of spaghetti thrown on the plot. And it can be really complicated. Even if there has been quite strong selection, we may not see it just by looking at the allele frequencies of the QTLs that affect our trait. And I should say it was a real honor to be introduced by Antoine Kremer, because he wrote a series of papers that really helped me understand this problem when I was starting my postdoc. So one way that evolutionary biologists have tried to make sense of this kind of data is to sort of separate out all of these allele frequency differences by their phenotypic effects. So if we color all of these lines by which alleles are more common in French populations versus, or which alleles were the tall alleles more common in French populations, and color things by which allele has the taller allele more common in Sardinian populations, we start to see that while these patterns are fairly complex, there are more cases where the tall allele is more common in French populations than in the opposite. And this is a signal potentially of adaptation for height in these two populations. But I'll get back to some of the caveats for this approach. And the way, so based on this idea though, the idea you can start to create tests of adaptation, which not only don't involve just staring at this plot and counting lines, but instead use a more comprehensive modeling approach, where first you find loci associated with height and use their effect sizes using a GWAS, and then combine these effect sizes with the allele frequencies in a set of additional populations, essentially by summing across all of your alleles, combining the effect size by the frequency to generate a polygenic score, and then testing for selection on these polygenic scores, which are themselves summaries of your allele frequencies across different populations. And so this approach was used by my colleagues Jeremy Berg and Graham Coop to look at human height. And so this is a fairly complicated plot, but the main thing to take away is on the x-axis, we have a bunch of different populations. On the y-axis, we have this polygenic score for height. Each of these dots is a different population. And for these very large dots, what we see is there's a very strong signal of selection that caused these height scores to be very different from the mean. And this is stronger than their model could explain would be due to genetic drift. And these scores don't necessarily correspond with height. What they're really telling us is that in these populations, tall alleles are at higher frequency. And at these populations, the tall alleles are at lower frequency. So as a postdoc, when I started, I was really interested in trying to use these methods and apply them to maize to try to detect polygenic adaptation. And the reason I wanted to do this in part is because maize is a really cool system for thinking about adaptation. It was initially domesticated in Mexico and has since spread worldwide and had to adapt to a wide range of environments. And this includes both natural environments. For example, highland maize has had to adapt to much more UV, pressure from UV rays, and also to shorter growing seasons. And so highland maize varieties have these hairs on their stems and also flower much faster. There's also a lot of interesting adaptation that's occurred due to human domestication or human selective pressures. For example, in Hala, in sort of Western Mexico, there's a variety of maize that's been selected to have really long ears. I can't show you in the screen, but imagine things that are like 40 and 50 centimeters long. And this is part of a sort of cultural adaptation. But what we end up seeing is quite a lot of phenotypic variation in maize that could be due to a number of different selective pressures. So first, I'll talk a bit about detecting adaptation in European maize, which is a great coincidence since this is a European consortium that I'm talking to. But in fact, maize in Europe has quite an interesting history. There were multiple introductions of maize to Europe quite early on during the time that Europeans began going to North America and South America. And maize spread quite rapidly. For example, there are these really beautiful paintings of maize and frescoes in Rome from the early 1500s that show that it has quite a long history on the continent. And so presumably it's had to adapt to quite a wide range of environments that differ a lot from its native environments. And so in particular today, I'll first start by talking about a set of European maize varieties that are from all of these locations here in Europe. So there were two main issues we came across when we first started trying to apply these polygenic adaptation methods to maize. And the first is that the lines in maize that we have don't always fall into clear populations. And so, for example, again, in these human examples, they're able to use these human populations. But in maize genotyping panels, we often see, if each of these points is a different maize line on genetic principal components. And while we can assign some of these lines to specific populations like tropical or stiff stock and non-stiff stock, which are two major heterotic breeding groups, we see a lot of lines that don't fall into clear populations. And because these are inbred lines, we can't really make the assumption that even the things that we call populations are sort of what we think of as populations. And so the way we got around this was by, instead of looking for differences between populations, we decided to develop an approach to look for selection along these genetic principal components. And so I'll illustrate that with a cartoon example. Imagine we have three populations here that are there are different subpopulations of species that follow this relatedness pattern. We could imagine that our first principal component, and I say imagine, but in fact, this is what we get when we simulate the data following this tree. We can see that the first principal component separates out this green population from these two blue populations. And the second genetic principal component separates out these two blue populations from each other. So by looking for divergence along principal components, we're sort of getting a more flexible way to think about genetic variation across a complex set of related populations. Next, the second issue is something that was a problem for maize, but also maybe a problem for a lot of other plant species where we want to detect adaptation. And that is that the GWAS that we do to identify loci to make these polygenic scores have been done in a species-wide sample. And so we can plot out the information we have for our European maize populations and our GWAS panel, look at them along principal components, and see that there's quite a lot of overlap between our GWAS panel and this maize panel. And so this is an issue in a number of plant systems where GWASs have been done in these range-wide structured populations. And so I'll show you first that this is definitely a problem using some neutral simulations. So we did simulations using our genetic information from maize, where we did GWAS in a set of North American lines, inbred lines, where we had randomly chosen neutral sites and assigned random effect sizes to these sites to create simulated traits that should be evolving neutrally because we've chosen neutral alleles. We used our GWAS on simulated traits to construct polygenic scores for our European maize varieties, and then tested for selection using these polygenic scores. And just to remind you, the polygenic score is a sum of our effect sizes and our allele frequencies across all of the loci that we found in the GWAS. And so this is what we found when we did this test for a number of simulations. On the x-axis is all of the principal components that we were looking at, and the y-axis shows the proportion of significant tests. Since we were simulating things that shouldn't be evolving, we would expect that we would see about 5% of our tests showing up at a p-value below 0.05. But in fact, we saw much more signals of selection, which we shouldn't have seen since these were neutral simulations. And so I'll tell you why I think this is happening. So we can think about now looking at our European lines, where we want to test for selection, looking along the genetic principal component 1, and at our simulated traits. And we see that there's no relationship between where things fall on the genetic principal component and our simulated trait, consistent with neutrality here. But when we start to look at what happens to our polygenic score, we see that we end up seeing this negative correlation between our principal component, or essentially spreading out our differently related individuals, and our polygenic score that's based on this GWAS. So something about constructing the polygenic score has contributed a bias to this estimation. And so my intuition for what's happening is that because the GWAS is itself controlling for population structure with a kinship matrix, we're biased towards finding alleles that have non-neutral allele frequency distributions across our population. And that bias gets propagated into our polygenic scores. But in reality, what this means is we might detect signals of selection using these methods from humans when we apply them to plant species, and that these would be false signals of polygenic adaptation. So we constructed a different test based on the multivariate normal, the conditional multivariate normal, that should incorporate these biases into our neutral expectation for what our polygenic scores look like under neutrality. And we showed that when we applied this to our simulations, we then saw no evidence of false positive signatures of selection. So now I'm showing you the proportion of significant tests, and they generally fall around 0.05, which is what we expect for a p-value of 0.05. And if you're interested in how this test works, I'm happy to talk about it later, or you can see it in the paper, but I don't have time to go into it now. So once we ran this test, we then saw that we're not able to find any evidence of polygenic adaptation in these European maize populations. So this is a heat map with all of the principal components that we looked at and all of our traits colored by the significance of the test for selection. And so while we do see some evidence of selection, none of it survives false positive correction. And so this is ultimately a little disappointing in that we seem to have removed any power by creating something that was robust to the false discovery problem. We've lost our ability, or we perhaps have eroded our ability to detect selection, at least in this situation. Or maybe that there's not adaptation at all. We don't really know. So that is the story with European maize and what we found. But the main takeaway here is just that there is a problem sometimes with taking these methods from humans and bringing them over to a different species. And I should note that even though I emphasize that issue, that the structure has also been found to be a huge problem in human polygenic adaptation studies also. I think Jonathan Pritchard talked about this last week or during the last version of this symposium. And these papers, if you haven't read them, are really wonderful at explaining some of the additional issues with population structure and GWAS. Okay, however, there are some big advantages to working in plants over humans. And one of the big ones is that we can actually measure traits in common garden experiments where we don't have to worry about there being different environmental inputs across different genotypes as much as we would in a human GWAS. And so this means, in a way, we don't need to make polygenic scores always. We can use the phenotypes that we have as a pretty good measure of the genetic variation that's present in these different populations. And so next I'll tell you about an approach we developed to try to skip this polygenic score test and just look at selection on phenotypes themselves and use that to try to understand the role of adaptation in shaping genetic variation. So to back up a bit, in order to detect adaptation shaping phenotypic variation, we need to know about two key things. First, we need to know that there's genetic variation in traits between different populations. And so in this cartoon example, we can imagine in a common garden, we grow individuals from population A and individuals from population B. See that individuals from population B are bigger than the individuals in population A. And then that tells us there's some interesting genetic variation here to explore. The next thing we need to know is relatedness information. Because if our two populations are quite distantly related from each other, we might not be that surprised to see that they have quite different phenotypes. Because if phenotypes drift neutrally, there's been a lot of time for that drift to occur. And so this is why relatedness is important to know. If our populations are much more closely related to each other, there's been less time for the phenotypes to drift. And so we might be more surprised to see these differences occurring to drift, and more likely to then say we need selection to explain these differences in phenotypes. And so this is why understanding genetic variation and relatedness is really key for identifying adaptation in phenotypes. And so this approach and this intuition has been changed into some really nice approaches called QST-FST that test for excess trait divergence compared to expectations based on neutral genetic variation. And so the idea is that QST measures population differentiation in phenotypes between populations, and FST tells you how much differentiation you'd expect based on drift, using neutral genetic sequence variation to sort of build that estimator. And so these approaches have been used for the last few decades to identify cases of adaptive trait divergence between populations. So we were interested if we could try to combine the principal component approach I told you about already with QST-FST to try to detect selection on phenotypes alone instead of constructing polygenic scores. And so I'll walk you through how we do that, continuing on with our cartoon capsella plants. So imagine we have our two populations of small and large plants arranged along the first genetic principal component that we've made using SNPs from these individuals. And so we can see that individuals from our large population are closer together because they're more closely related to each other. And individuals from the small population are close together because they're closely related to each other. Next, we can start to construct a correlation between, or look at the relationship between our principal component and our phenotype here, which is size, and see that our large population is larger than the small population. And that creates this linear relationship between the principal component and the phenotype. And this could be consistent with drift. And maybe the large population is just larger because of neutral genetic drift. And so what we need to do is to build a model for how much difference between our populations we expect to see just due to drift alone. And so we can imagine thinking about the slope of this relationship as sort of what we're trying to model. And under neutral drift, we could expect that slope could go either way because drift is equally likely to increase or decrease population level phenotypes. And so we can build a model then of what we expect this slope to be. And so we call the slope QM, which is the slope of the trait plotted against principal component M. And under neutrality, we model this as having a mean of 0, since drift is equally likely to affect the phenotype in either way. And then the variance here depends on the amount of relatedness explained by the principal component, or this is the eigenvalue that goes with that eigenvector. And then also VA, or additive genetic variation. And one really nice thing about using principal components is that we can use a subset of the principal components to estimate VA, and then look at selection on different principal components. And so I should note that this is really a rearrangement of the animal model in some sense. And so that's kind of where a lot of this math comes from. Okay, so now that I've explained the neutral model, we can imagine that in fact, selection has really increased the size of the individuals in this population. And so we now see a slope that's much larger than we can explain under neutrality. And this would be our signal of adaptation, shaping trait variation here. And so we can rearrange the equation I just showed you into a test for selection, creating trait divergence between populations or across our principal component, and then model this as following an F distribution. And so this is the test statistic that we're actually using to test for selection. Okay, so now I'll walk you through how this works, again with maize. So in this case, we're talking about essentially our GWAS panel that's been phenotyped for 22 traits. We have whole genome sequence data, and this comes from 240 domesticated inbred maize lines. And just to show you the genetic landscape here, in this sample, principal component 1 separates out our tropical from our stiff stalk lines and some of our other temperate lines. So maize was domesticated in the tropics and then spread up north into North America into temperate regions. And that's described on principal component 1. And then principal component 2 sort of separates out our stiff stalk from non-stiff stalk lines. And these are the two major heterotic breeding groups that have been used in North American breeding in the past. And we also have a few popcorns and sweet corns that then get separated out by some of the lower principal components. And so this, I'm going to walk you through what the results of this test look like for this panel. So I'm showing you now on the x-axis, principal component 1. On the y-axis, we have kernel number, which is a phenotypic trait in maize. And these dotted blue lines are showing you what our model's neutral expectation is for the relationship between the principal component and kernel number. So if our regression line essentially falls in the middle here of this sort of hourglass shape, that's indicative of neutral adaptation. If it falls outside of it, that would tell us that selection is needed to explain the variation that we see. And so for kernel number, what we see is that there is some relationship. So the stiff stalk lines have more kernels on average than our tropical lines. So we see right here this slope of the relationship, but it's well within our neutral expectation. And so this tells us that kernel number adaptation along principal, or kernel number variation along principal component 1 can be explained by drift. However, we can also look at another trait like flowering time. Again, we have principal component 1 on the x-axis and flowering time on the y-axis. These are our neutral expectations. And here we see a different pattern. The slope of the relationship between these two values is outside of our neutral expectation. That slope is shown here with the dark blue line. And so in some sense, this is unsurprising. We knew that tropical lines flowered a lot later than our temperate lines because they grow in areas with much longer growing seasons. However, what this test now tells us is that this difference is greater than we could explain due to neutral genetic drift alone, and so it's likely due to adaptation and selection. And so this is the new, that's what it's, that's the main conclusion we can get from this. And so one nice thing about this test is we can now apply it systematically to all of the phenotypes we have measured across a range of principal components. And we end up getting something that looks like this heat map, where on the x-axis we have principal components. On the y-axis, we have phenotypes, and they're colored in by the significance of the test for adaptation. And then those white circles tell you that the test has passed a false discovery rate control for multiple testing. And so what we can take away from this is that just by going through and putting in this data we had from these GWAS studies, we can detect adaptation for flowering time, both male and female flowering time, and no number below ear, along a number of principal components. We've also looked at, to live up to my title, we've also looked at this at gene expression, at the gene expression level. This was work that was led by a really wonderful undergraduate in the lab, Jennifer Blanc. She took gene expression data from 208 of these maize lines from 7 different tissues, and that includes different parts of the leaf, developing embryo, and kernels, as well as leaves that were collected during the day and the night. And so what Jennifer did was she took the test that I just told you about and applied it now to gene expression levels. And so the plot looks a bit different because she made these, but essentially what we now have is principal components on the x-axis still, and on the y-axis, instead of a phenotype that we could measure, we have gene expression. And so in this example, we see that the gene expression divergence along principal component 5 is greater than we could explain just due to genetic drift. And so the cool thing about this approach is we can look at gene expression data. Since it's RNA-seq, we can look at gene expression data for tens of thousands of genes across these 7 tissues and try to understand in general what role adaptation has played in shaping this variation. And so this is a heat map of what Jennifer found, which is that we can look, she looked across 5 principal components and these 7 different tissues and found the strongest signatures of selection. So the most genes under selection along this principal component 5 for daytime expression level. And so this principal component 5, just to zoom back, is sort of separating out a bunch of our non-stiff stock lines that are one of the major breeding groups used in temperate maize breeding. And so if you're interested in this, she has a really cool preprint out, and she delved into a bit more sort of trying to understand the selective pressures that might be contributing to these signals of adaptation. To sum up this section, I'll just add that the cool thing I think about this approach is that we could apply it to most GWAS panels where people have genotyped and phenotyped large numbers of individuals to systematically get a view of what role adaptation is playing in shaping trait variation. And in particular, I'm really interested in extending this approach to look at selection on plasticity, and I've been doing that using Arabidopsis flowering time data. And if you're interested in applying this data to your own, or this method to your own data, there is an R package that you can use. And I'm also happy if you want to contact me to talk about it. Okay, so for the last 15 minutes, I'm going to change gears and tell you a bit about thinking about adaptation in the present. So how does, and the question in particular I'm interested in is thinking about how plasticity contributes to adaptation to herbicides in weedy species that plague domesticated fields. And so I'll tell you first a bit about why I think this is an interesting question. So as evolutionary plant geneticists, we're all quite familiar, I think, with the idea that traits are shaped by genetics and by the environment. And this was has been shown for decades and decades now, including these nice demonstrations from Klausenkek and Heisey, who grew plants in different environments, from different environments, and then put them into different environments, and showed that phenotype really depended both on genotype and bioenvironment, and you get really complicated interactions between the environment and the genotype in shaping phenotypes. And so this G by E, or genotype by environmental interactions, can be caused by genetic variation for plastic responses to the environment. And so understanding why they exist really requires understanding when and where environmental responses can be adaptive. So I like to illustrate this with the shade avoidance pathway, which is a really nice example of adaptive plasticity in plants. And so the way this works is that when you grow a plant in the shade, there's often a classic response where the plants will grow taller to try to escape their neighbors and get sun so that they continue to grow and reproduce. And so this response is generally thought of as being adaptive. However, these responses can also be maladaptive. Imagine you're a plant living under a big tree. You can grow as tall as you want. You're a little herb and you'll never grow taller than the tree that's shading you. And so in fact, some forest floor species have lost this pathway because that plastic response is no longer beneficial. And so understanding the effects of plastic responses on an adaptation is really important for thinking about the maintenance of G by E. And so there are some really classic examples or studies that have shown that plasticity can be adaptive. And so this is a really lovely example from Susan Dudley and Annie Schmidt, where they used experimental manipulations to actually show that plasticity was adaptive. They took plants, and for some of them, they elicited a shade response, and for some of them, they let them elicit a sun response. And then they transplanted these plants into different environments, a high density where the plants experience a lot of shade, and a low density area. And they showed that the individuals who had elicited the shade response had higher fitness in the high density areas, but lower fitness in the low density environment. And so this is indicative that this shade avoidance response can be adaptive in some environments, but not in others. However, many of the traits where we might be interested in thinking about the role of plasticity or environmental responses are not things that can be easily elicited and then held still so that we can then do these manipulative experiments. And so I'm really interested in approaches for trying to understand the effects of plastic responses and some of these traits where we can't manipulate them as easily. And so today I'll tell you about a project I've done in collaboration with Regina Balcom and Megan Van Atten at the University of Michigan, looking at pesticide resistance in morning glory. The morning glory, while it's beautiful, I have it growing in my garden. It's also a fairly important weed species. And so this is an experiment that Gina and Megan did using a natural population of morning glories. So they took this population and they selected for resistance to glyphosate, which is one of the major herbicides used on morning glory, for 3 generations. And then they also did a control where they just randomly picked individuals to get individuals that had been evolving in a lab environment, but not selected for glyphosate resistance. And so they ended up with 2 populations. This population showed glyphosate resistance and this population did not. Then they looked for differences in gene expression between individuals that had been sprayed and not sprayed with glyphosate treatment, with glyphosate. And I should say this is part of a larger experiment that's really cool. There's a preprint up from Megan and Gina about it. And I'm just going to tell you about the piece of the data that we used to answer this question about plasticity. And so the cool thing about this setup is we have individuals that have been selected for resistance, and we have information about the plastic responses that individuals show when they're treated with the selective agent. And so we can compare the expression of these 3 different groups to try to understand the effects of plastic responses. So in particular, we're interested in thinking about the plastic expression responses that occur between plants that have been sprayed with herbicide and plants that haven't been sprayed with herbicide. And I should note that all of this expression was measured in leaf tissue. We also have the evolved responses and expression between control plants and plants that were selected for resistance. And so we can use these 2 comparisons to try to think about whether plastic responses are adaptive. And so I'll walk you through the results and how we did this. We had expression data for 27,000 genes, and I've plotted each of these points as 1 gene, and I've plotted out on a log scale. The x-axis shows the plastic response. So points up here had greater expression after being sprayed than genes down here. And on the y-axis is the evolved response. So genes up here had greater expression after selection for resistance. Genes down here had lower expression after selection for resistance. And so overall, we found that 11,000 genes had significant plastic responses to the glyphosate spray, either positive or negative. We also saw that 299 genes showed significant evolved responses to being selected. So they had either higher or lower expression in the resistant lines compared to the control lines. And finally, there were 94 genes that had both significant evolved and plastic responses. And that's the sample we're going to use to try to understand whether the the potential adaptive effects of plastic responses. And so this is how we actually did the test. You can imagine seeing a gene that has a negative plastic response to being sprayed and a positive evolved response. So this means that when you spray the plant with herbicide, the expression does the opposite of what it does when you select for herbicide resistance. And so we take this as an example of maladaptive plasticity because plasticity goes in the opposite direction as selection. Alternatively, we could imagine a different gene where both spraying the plant with herbicide and selecting for herbicide resistance increases expression. And so this pattern is consistent with adaptive plasticity because the plastic response is going in the same direction as the evolved response after selection. And so when we look at all 94 genes where we can test for this pattern, we see evidence of both adaptive and maladaptive plasticity. So in some genes, the plastic response goes the same direction as selection, and in some genes it goes the opposite direction. But for more genes, there's evidence of adaptive plasticity in the sense that the gene expression is changing in the same direction as during spray as it did during selection. And so we took this as evidence that in general, there's stronger patterns of adaptive plasticity than maladaptive plasticity. And I was super surprised to find this because I'm not the first person to do this experiment. There's a really nice example from Cameron Gallenber's group looking at guppy brain gene expression. And so again, this is the exact same kind of plot I showed you, only with guppies that have evolved in different predation environments, comparing the evolved responses to predation to the plastic responses. And in this case, for guppies, there was really strong evidence of maladaptive plasticity, and that if you stick guppies in a different predation environment, their brain gene expression does the opposite thing that it does when you evolve them in that environment. Similarly, one of my colleagues from grad school, Yuheng Huang, did the same kind of experiment looking at Drosophila that had been evolved on cadmium and salt diets, compared the evolved response to the plastic response. And he also found really strong evidence of maladaptive plasticity. And so this leads me with the final question for the section that we're trying to figure out and sort of would motivate future work in the system, which is why do we see so much adaptive plasticity in morning glory gene expression relative to these other case studies where this has been examined? And it could be that, you know, this population came from a farm that had experienced some amount of herbicide spray already. So maybe this response has occurred due to adaptation that's already occurred. It could be that these adaptive responses are related to other abiotic stress responses that morning glory has had to adapt to for a long time. Or there just could be something fundamentally different between plants and animals. We don't really know, but I'm really excited to find out in the future. Okay, so to sum up what I've told you about today, I've told you two stories about polygenic adaptation in plants. The first thinking about how adaptation shapes quantitative trait variation within present day quantitative trait variation, and sort of thinking about not only the answer to that question, but how do we even ask that question using the data that's available to us. And next, I told you about how plasticity might contribute to adaptation to herbicides in a common weedy species. And I didn't get to talk about the future, but that's one of the reasons that I really feel motivated by these questions, is sort of thinking about how we can better understand adaptation and use that information to predict future responses to changing environments. So I have a lot of people to thank for the work I told you about today, in particular, my collaborators in maize, Jeremy, Graham, Jeff, and Jennifer, who were really wonderful to work with on the system, as well as the Rossi Barrett and Coop labs. And I'd like to thank my collaborators at the University of Michigan and Michigan State, and along with Alex Harkis and Jim Liebensnack for work in this morning glory system. And also thank you to you all so much for listening. I'm really excited to talk to you further.
Thank you very much, Emily, for this excellent talk. Thank you so much. We are going to open to questions now. Before we do, I wanted to just briefly explain how we are going to go about it. And you have a participants list on your right when you click on the participants button at the bottom bar of your Zoom. So under the list of participants, there is a button called raise hand. And so I'm just asking to all of you who want to ask any questions to raise your hand, and then we will take the questions one by one. There is also a chat box here, so in case you prefer to write your question, you can also go for that option and just write it in the chat. We've already got the first question from Thomas Bataillon. I don't know if I'm pronouncing it right. If you can just introduce yourself and say where you're coming from, Thomas.
Okay, thanks a lot. So my name is Thomas Bataillon. I'm currently based at the Bioinformatics Research Center in Aarhus, in Denmark. And I guess I've been interested for quite a few years by modeling, departure from neutrality and polygenic selection. So I was very interested by the first part of your talk. And I was wondering, what was your viewpoint about how to test for selective neutrality, both with single locus and polygenic selection? Because I have a feeling that throughout the method that you've shown us, the procedure is always in my mind the same. We formulate a neutral model, and then we reject the neutral model, and then we reject it, we say, oh, it's selection. But I think it's been very clear, at least in the recent years, especially with GWAS studies that have massive amounts of data, is that even tiny misspecification of the neutral model can actually lead to situations where there will be no selection, but we just will reject the neutral model because we have misspecified it. And I was wondering, so I thought that the work you presented on how to predict the sort of the neutral envelope for the slope versus PC was very elegant, but I was wondering to which extent you would fear that you can run into similar things. Like, is it really a neutral variation around the slope that is adequately described by the Gaussian that you're describing, or is there more to it? And
yeah, I don't know if I can satisfactorily answer that question. It's a really interesting point. I do think that there are some ways that we constructed the test to try to be robust to that, in the sense that we modeled sort of what role environmental variation in our phenotype would play and show that it's relatively conservative to that, since environmental variation will really just increase that envelope. But yeah, I think we depend on certainly being able to model, sort of use the fact that we're depending on the fact that the neutral genetic variation is telling us something useful about what neutral phenotypic variation would do. So yeah, I don't know how to answer it, but I think, you know, it's correct to be concerned that this has been a problem with this kind of work.
Thanks. I think it's a very elegant method, but I was just wondering a little bit, as we have a lot of data, it was a little bit the approach of Berg and Koop that you were showing also earlier without going into the details. I feel that it's very elegant work, but it always relies on relatively strong assumptions about how the genetic drift is modeled and how it shapes the test statistics that we're looking at. Mm-hmm. And it seems to me that it was perfectly fine when we had 30 SNPs or 100 SNPs, but now when we have 500,000 SNPs, even the most insidious small deviation, they create huge signals. And it was very obvious in the papers that you presented about the problem with structure in human height, for instance.
Yeah. Do you have idea, like, would it make more sense to move to sort of a multi-hypothesis testing framework, or like, what do you think the right approach is?
Yeah. But I don't want to monopolize the space for more questions. Thanks again for a really nice seminar.
Thanks.
Thanks, Emily. I would like to ask Antoine if he has any comments at this point.
Yes. Well, still on the same issue that was raised by Thomas, I wanted to ask about the polygenic score. I mean, I wonder if, I mean, there are two components in it. There are the gene effects, of course, and the allelic frequencies. Are those estimated in the same experiments, or are those coming from different, I mean, experimental setup? But this may also be a reason that you get not the signal of adaptation or.
Yeah, the way this has worked for those kind of polygenic score methods is that there's a GWAS done in one population to estimate the effect sizes. And then there's the test is done in like a different population. And so sometimes those could even be quite different sequencing data. Like the European maize I talked about comes from a SNP chip, whereas the sequencing data from the GWAS comes from short read sequencing. I'm not sure if that's helpful for answering the question, but they do. Ideally, they would be very independent, because that would solve some of the issues that come with this shared structure problem.
Okay. And then there's no, you don't include interaction between, I mean, there's no epistasis. Is this included, or?
Yeah, right now the model does not include epistasis. So we're modeling phenotypes as additive combinations of allelic effects. And so epistasis would certainly change violate some of the assumptions of the model.
Okay. Mm-hmm.
We've got another question from Christian. Christian Rödstad.
Yeah, thank you for the nice talk. I was wondering about the second part. You delivered an explanation why you have adaptive plasticity, but all the other examples had actually a lot of maladaptive plasticity. I was wondering why is this the case? Why should a plastic response be like in contrast to the one you should actually evolve to? What is the mechanism behind that?
I think that's a really good question. My assumption is that, in some of these cases, when you put an organism in a new environment, you're exposing variation that's never been under selection before, if the environment's truly new. And so in that case, it's not that surprising to see maladaptive plasticity, because you're really just revealing a lot of variation that's been unselected in the past. So that's my interpretation of it. And so that's why I think for this morning glory example, it must be that we're picking up on responses that are responses to other things also that maybe the plants have experienced. But it could be that, I guess it's just chance what happens when you put something in a new environment, potentially.
Thanks, Emily. We've got another question from Hernan. Hernan, can you please introduce yourself?
Yes, hello. My name is Hernan, and I'm a PhD student at the Swedish University of Agricultural Sciences in Uppsala, in Sweden, and I'm working with Norway spruce. Thank you so much, Emily, for your presentation. Really, really nice work. And I would like to come back to the point that we were speaking about before, about the polygenic score used in another population different from the one that you used to calculate the effect sizes of those SNPs. And maybe I missed it on your presentation, but is it the case that you can also use the traits that you measure in the different population, not the one that you use for the GWAS? And did it match the signature sort of adaptive selections on those traits in these new populations with the polygenic scores from those traits in the population used? It's such complicated, but I hope you follow. But in this way, you could, in a way, validate that the polygenic score of that trait is indeed under adaptive selection, because the trait in this other population also follows this pattern.
Yeah, I think there's two issues related to what you're saying. So you're talking about how we have the GWAS where we make the polygenic, where we estimate effect sizes, and then the area where we test for selection, in this case, like the European maize. Yeah. And so the first case is if we have phenotypes. So in this case, we did not have phenotypes for these lines, so we really had no way to validate what we found, and that would be really nice if we did. However, I think in that case, it might also be tricky depending on where they were phenotyped. So there's some nice work from the human side of this, looking at the idea that, if your environment is correlated with genotype, that the phenotypes you observe might not actually reflect your polygenic scores for different reasons. Yeah. And I'm not I can't really explain that right now, but I would definitely suggest looking at that. But yeah, in this case, we did not have the ability to validate that, and that would be useful and interesting, but potentially a problem if things are measured in different environments.
Yeah, thank you.
Thanks, Emily. Are there any other questions? I've got comments here, Emily, people saying very nice talk, great talk, compliments for you. So I'm just reading these comments here for you. I think Antoine would like to say something.
Yeah, I'm jumping in again. I'm coming back to the issue of adaptive plasticity. Now, we are wording this a little bit differently in trees. We use the term counter-gradient variation and co-gradient. When, for example, we have genetic values of population that form a cline with an environmental gradient, and we see phenotypic values going in a different direction for the same trait when they are measured in situ. So there are certain series of threads that follow, for example, phenological threads. In many species follow counter-gradient variations. But in most cases, we have co-gradients, so which means that probably while there is plasticity is likely to be adaptive. So our explanation, our interpretation, but this is very rough, was that there might be some internal, let's say, trade-offs between different threads that make that they may vary differently at the phenotypic level than at the genetic level. But in any case, there is cases in trees where we do have differences between the adaptive gradient, genetic gradient, and the phenotypic gradient. So this was a comment, and a further question. In your first part, you showed the in maize these experiments, and there was an illustration of the genetic variation of various threads, and there's still immense variation, genetic variation. I was wondering if you do find any correlation between the level of variation that you observe and the genetic architecture of the threads in those species. I mean, in terms of number of QTLs or whatever, or number. Mm-hmm. Do you see anything?
Yeah, that's a good question. I haven't looked at that directly at all, but trying to think, like I can say anecdotally, you know, for flowering time, there's a lot of variation and a lot of small effect QTLs in maize. What would you expect to see?
Well, I've no, at first sight, I would expect a correlation with the level of architecture, but there's no, when you go into detail to it, there's no very genetic reasons for it to being so. So I was just wondering if at the experimental level one can see a correlation or not.
Yeah, I don't know for sure, but I'm sure somebody knows in maize the answer to this.
Maybe somebody in the room?
Yeah.
Okay, so are there any other questions for Emily?
Well, I may ask a question. I raised the same question to Jonathan last week. And we are all doing our experiments in as you said, in common garden, experiments to study adaptation and so. But we are wondering what's going on in situ, in nature. And this is really where selection, natural selection is going on. And I wonder how much you can, you can take conclusions from controlled experiments and even experimental selections, selective selection experiments to conclude on evolution and adaptation under natural conditions.
That's a really good point. And I think, well, one thing I like about working in maize is it might be a case where we are doing a good job of mimicking its like natural environment when we grow it in the field, because depending on what we're thinking about, we're growing when these things are phenotyped in experimental fields across, the phenotypes come from some of the same kinds of conditionings that breeders are using to breed maize. And so that alleviates it a bit, although adds another question about, what does adaptation in maize tell us about adaptation in natural systems? But I do think that's a very good point for like morning glory herbicide resistance. So people have been studying sort of pesticide resistance as a model of adaptation for quite a long time. And in the mid part of the last century, I think people were looking at it in Drosophila, and they found very different responses when they compared experimental evolution to sort of natural populations of resistant flies. And so I think that's something that, I know the Balcom Lab is currently working on looking at sort of natural populations of resistant morning glories alongside the experimental evolution. And I think that's going to be really important because it's really interesting, either way, if we see that there's similar genetic bases to resistance to experimental evolution as to natural populations, that's really cool. And if there's not, that's pretty interesting also. And so I'm sort of excited to see how that turns out.
Okay, this is the last call. Now is your last chance to ask any questions to Emily. But then also, if you have any comments or later on, we can always pass them on, and you can write to evoltree@efi.int. And we also welcome to engage on Twitter at evoltreenw. And we are very grateful for you, Emily, today for your excellent talk. So thank you so much for joining us for this series. And we will be reconvening again in 2 weeks. So for the third episode. But for today, that's it. And thank you very much to the online audience who joined us. And see you in the next episode.
Great. Thank you so much, everyone.
Thank you.
Hope everyone has a good rest of your day.
Thank you very much, Emily.
Thanks.